Hello, chemists and biochemists. In this video, what I'd like to talk to you about is enzyme regulation. Now, that discussion is not going to include enzyme regulation by inhibitors because I want to talk about that completely separately. But with that in mind, with regulation in mind, there are a couple things that I want to talk to you about. In particular, the ways in which enzymes can be regulated can be classified in a couple of different ways. There are regulations that are considered to be covalent, non-covalent, then each of those can be classified as reversible or irreversible. So let's first talk about our non-covalent regulations. In general, your non-covalent regulations are probably going to be reversible. Now, an example of a non-covalent regulation would be something like an allosteric effector. So think about a molecule, or think about an enzyme, which I'm just going to label E down here. And let's think about something, let's say molecule, which I'm going to label X. Molecule X. Let's say molecule X binds to our enzyme. Let's say that there's some hydrostatic and hydrophobic interactions that stabilize this complex right here. E mol X, which I'm going to label there. This right here, this combination of these two molecules, well, it could be activated. It could also be inactivated. Now, the key is regulation doesn't necessarily mean preventing activity. It's just kind of turning it on or turning it off. So if binding of X causes E, our enzyme, to change its shape so that it can now bind, let's say our natural substrate is this little triangle-shaped molecule. Let's say when E and X get together, you form something that looks like this. EX. Well, E, from its initial state, where it kind of, and this is just for the illustration, it kind of had a rounded opening. Whereas when X bound to it, now all of a sudden it has a very great binding site for its natural substrate, S. Okay, so an allosteric effector is going to change the shape of the protein, generally through non-covalent interactions like ionic or hydrophobic interactions. Okay, now let's take this in the direction of, or let's now talk about covalent interactions and covalent regulation. Well, covalent regulation opens up this whole, this very large range of modifications to proteins or enzyme modifications. Which are referred to as post-translational modifications, or as I'm going to call them PTMs. And now, the key here is it's post-translational. So any modification to an enzyme after translation would be classified as a post-translational modification. Now, for our covalent modifications, what we're talking about is, well, adding, in this case, or what I want to narrow focus in on, is adding a small molecule to the enzyme. And I say adding because we're going to have a covalent linkage between our enzyme and this molecule. And a couple of kind of classical and nice examples would be phosphorylation. Phosphorylation is one. Another example of a covalent modification would be, I'll go with acetylation. as well as methylation. Now, these right here are reversible. A phosphorylation is going to be a phosphate group. So you generally see it depicted as OPO3 added to an amino acid like serine, threonine, histidine, or tyrosine. Now, that phosphate group can be added to that amino acid, keeping in mind this amino acid is found within our entire protein. It can be added to there and it can be removed from there. I'm just going to add that -2 charge because that OPO3 is going to bring a charge along with it. So that group, that phosphate group can be added, it can be removed. In some cases, phosphorylation leads to activation of an enzyme. In other instances, phosphorylation leads to, well, a depression in activity or maybe a reduction in activity. Now, acetylation is going to be a acetyl group, so added to an amino acid, specifically a lysine at the N termini of a polypeptide sequence. So I'll just add in N term. So you'd be adding in this case, you add an acetyl group to a positively charged amino acid like lysine, and basically take it from something that's positively charged to something that is effectively not going to have a charge and is considerably less polar. Methylation is another example of a reversible covalent modification, and that's where you add a CH3 to a glutamic acid residue. Keep in mind, a glutamic acid is going to be a negatively charged residue. Adding a methyl group, well, you're going to take something that's polar and make it considerably less polar. Okay, so these are all reversible modifications. Those groups can be added, they can be removed. Now, there are also covalent modifications that are irreversible. An example of this would be something like in the case of a protease or proteases. These are enzymes that what they like to do is they like to cut peptide bonds. And what they do as they cut these peptide bonds is they effectively kill an enzyme. And so a protease is an enzyme that targets other enzymes and other proteins, which might sound a little bit strange or cannibalistic, I guess. But these enzymes, we do not want them to be active all the time. So what they go through is they are processed, they are translated into something known as a zymogen, which is an inactive protease. A zymogen will then be proteolytically cleaved, it will be cut, and as it, well, it's cut by another enzyme or another protease. It will then go on to cut itself to fully activate itself. Now, the thing is, these are peptide bonds that are broken. These peptide bonds can't be repaired, and they're not going to be repaired. So that's why this is an irreversible covalent modification, because we're breaking a covalent bond. It's not going to be repaired. So proteases are examples of covalent or irreversible covalent modifications. Phosphorylation, acetylation, and methylation are all examples of reversible covalent modifications. Then anything that non-covalently interacts and causes your enzyme to either stay in one shape or contort itself to make a different shape, that's known as an allosteric effector or an allosteric regulator. Now, there are other examples of allostery and allosteric proteins. For instance, hemoglobin is one. And based on the natural binding partner, well, you can classify allosteric regulators in a couple of different ways. But I hope this was helpful to look at enzyme regulation and how you can kind of classify different types of enzyme regulation, activity regulation. I hope this was helpful and hope you have a good one.